We are at the Institute of Macromolecular Chemistry, Czech Academy of Science. This is a big research institute devoted to different branches of polymer science, and the team which is involved in the EATRIS network is the Supramol team. This team, which is composed of two departments, is devoted to supramolecular systems. The Supramol teams contributes to the small molecule platform within the EATRIS network. We are unique in the EATRIS network in the point that we have equipment for both polymer synthesis, detailed physicochemical characterization, and different methodology to study the self-assembly dependent on external stimuli. Basically, our focus is from the application point of view, clinical applications view, is focused on oncological applications, infectious diseases, and diseases that are characterized by malfunction of essential elements metabolism, like Wilson's disease. From the, let's say, instrumental point of view, we have synthetic labs, we have liquid chromatographs, we have different scattering techniques to study self-assembly and disassembly dependent on external conditions, spectroscopical methods like time-resolved fluorescence spectroscopy, and also isothermal titration calorimetry, which can be very useful for studying interactions of the nanospecies with biological environment, for instance. Supramol team is focused in the pipeline on the beginning, so synthesis of these systems, physicochemical characterization, and in vitro studies in non-biological environment. Our expertise, we use our expertise for drug delivery systems for oncological and antibacterial applications. Then we also develop polymers to scavenge metal ions for treatment of hereditary diseases connected with malfunction of iron and copper metabolism, so hemochromatosis or Wilson's disease. We are also developing sensors for early detection of inflammation and infections, and we also develop different methodology of microfluidics for the preparation of nanoparticles, polymerosomes, and giant vesicles on chip. The collaboration with the other partners within the EATRIS network is based on the expertise of ours and of the colleagues. So we provide material, we design the materials and provide it for biological testing and for further use with the partners. For instance, we have collaborations with Charles University, with the First Medical Faculty, where we prepare the polymer and nanoparticulate systems which are tested on the First Medical Faculty or are tested in the Institute of Molecular and Translation Medicine in Olomouc. We also collaborate with the other institutes of the Czech Academy of Sciences. We also collaborate with companies, for instance, VH Pharma, or we also produce nanoparticles, for instance, for agriculture now, for the Agra company. And of course, we collaborate also with foreign institutions, mostly academic, but also companies. So, for instance, we have a good collaboration with CA Saclay in France, which is basically a research institute with the group of Eric Doris. Then we have a good collaboration with the University of Minnesota at Minneapolis with Professor Tim Lodge. We have a very extensive collaboration with Ghent University with two groups, Professor Hochenboom group and Professor van Vlieberge. Which is important for these collaborations is that we not only provide instruments, but also expertise behind. In the EATRIS network, there is a kind of, let's say, advertising of the technologies offered. So if somebody needs, some team needs expertise, they first go to EATRIS, and they pick up the team for which they need due to the expertise. And this is the networking role that the EATRIS has.